This video will describe the histology of the nervous system. The first slide we're going to look at is a nerve smear, sometimes called a motor nerve smear. And there are going to be two types of cells visible on the slide. The first cells are going to be neurons, and the other types of cells are going to be neuroglia. Neurons are the cells that actually conduct the electrical impulses, and neuroglia are all of the supporting cells. Now there are several types of neuroglia, but we're just going to lump them together into that one term. We are going to be looking for the soma, the axon, and the dendrites. Those are specific regions of the neuron. The soma is sometimes called the cell body. An axon is always going away from the neuron, and dendrites are going toward the cell body. When we look at a nerve, this is a bundle, a nerve is a bundle of axons. And so surrounding the entire nerve is going to be a membrane called the epineurium. And then we're going to have a fascicle, and that's going to be surrounded by a perineurium. And then we're going to have each individual axon surrounded by another connective tissue membrane called the endoneurium. And if you are in my class and you were there for the muscle lecture, and I used the example of a crayon box, it's the same idea. The full crayon box is the epineurium surrounding the whole box. Each individual, those smaller boxes, those smaller cardboard, that's the fascicle, and that cardboard around each smaller bundle is the perineurium, and then the paper around each crayon, it would be the endoneurium, if the crayon is the individual axon. We're also going to take a look at the spinal cord, a cross-section of the spinal cord. And we'll only look at one region, but you could look at cervical, thoracic, lumbar, sacral, different regions. But we're going to be looking for these different features. First, a central canal, an anterior median fissure, and a posterior median sulcus. Some references actually call them both sulcus, an anterior and posterior median sulcus, but technically there is a difference. A fissure is a deeper groove, and a sulcus is a narrow groove. We'll be identifying the anterior and posterior horns, the anterior gray commissure, and anterior, posterior, and lateral columns. For nerve endings and receptors, we're going to be looking at a Pacinian or laminated corpuscle. The fact that it has the word laminated refers to the fact that there are several different layers of connective tissue surrounding the actual nerve ending. If you remember in bone tissue, they are lamella, or the layers. So laminated just means layers of connective tissue. This is a type of mechanoreceptor, meaning that it is going to respond to mechanical changes in the skin, and it is located in the dermis portion of the skin, and it is responsible for detecting vibration. Another type of receptor that we're going to look at are the taste buds, and you probably know they're located on the tongue, but specifically on the surface of the tongue are little peg-like structures called papillae. Papilla is one, papillae would be plural, and on the sides of each of these papillae are these taste buds. And this is a type of chemical or chemoreceptor because the receptor responds to chemicals that are in the mouth, and so that is responsible for taste. I will be using slides from the Ohio State University Medical Center, and here's a list of those slides. Our first slide is just going to be the nerve smear, the neuron smear. This is actually taken from the spinal cord. And so I'm going to magnify this. And so all these really dark purple that we see are multipolar neurons. And all the little tiny purple dots that we see around the outside are going to be the neuroglia, or the supporting cells. Now, if I look at one multipolar neuron, multipolar because it has many different extensions coming off of the cell body, so it's multipolar. So the kind of star-shaped part in the middle is the soma, or the cell body, and I don't know, like that really dark spot in the middle, if you can see that, is the nucleus. then of all of these extensions, I'm really not going to be able to tell which one is the axon and which ones are the dendrites, but I'm just going to pick one. So if I say that this extension here is the axon, it's going to carry information away from the cell body. And then all these other extensions are going to be dendrites, and they're going to bring information into the cell body. So this is a multipolar neuron. There's a nucleus in the middle. These would all be the dendrites. There's one axon leading away, and then all these little purple dots that we see out here are different types of neuroglia. The next slide we'll look at is a cross-section of a nerve. And so as we can see here, this would be the nerve as a whole. And then I see a bunch of little individual, these are the fascicles inside. And then as I zoom in even farther, I'm going to be able to see that all of these little circles are the individual axons within the nerve. So going from the outside, largest to smallest, around the outside of a whole nerve, you're going to have the epineurium. Surrounding each fascicle, you're going to have the perineurium. And then as we zoom in, surrounding each individual axon is going to be a connective tissue covering called the endoneurium. And then the darker dots in the center of these is the axon itself. Okay. Then we want to take a look at the spinal cord. And I think we'll look at the cervical region. Yes, this is a good one. Okay. So the first thing that I look for is that I can tell the difference between gray matter and white matter. Remember that gray matter is going to be the cell bodies and unmyelinated axons, and white matter is going to have myelin on it, myelinated axons. So here in the center, kind of this almost butterfly shape, is going to be the white matter, and actually I can see a bunch of cell bodies here. And then surrounding the outside, I know it looks darker in this one, but this is actually the white matter around the outside. Here we have the central canal, kind of that hole in the middle, that's going to contain CSF, cerebrospinal fluid. The gray commissure, you'll notice that, so this is gray matter, kind of this butterfly shape, and there is some gray matter that crosses from the right to the left, and that area where it crosses from right to left is the gray commissure. Okay, so a few other features that we have. First of all, let's orient ourselves front to back, or anterior and posterior. This is a little bit backwards from some other slides that you'll see. This is actually the dorsal or the posterior portion, and this is going to be the anterior. So the anterior median fissure is this large or larger groove on this side, and then where this would be, there is a kind of a line right here. This would be the dorsal median sulcus because it's narrower. So it's not great, it's not a very visible, but it would be there. So if this is anterior, and remember that anterior is the same thing as saying ventral. So we also have sometimes called the anterior horn or ventral horn. That would be of the gray matter. Then we would have the anterior column. Anterior column is white matter, and so kind of from this area to the median fissure would be one anterior column, and then from the median fissure to this horn is the other anterior column. The lateral white columns are on the sides, that kind of makes sense there. And then this is going to be the posterior white column, so it's going to be from the median sulcus to the horn here. So this area is going to be the posterior column. Now then, that means that this area of the gray matter is the either called the dorsal horn or the posterior horn. So you'll hear it called both things. I think that's all of the different structures that you need to know for the spinal cord. All right, for the laminated corpuscle, we're actually going to look in the skin because that's where it's located. We said that it was deep to this in the dermis. And it's actually this structure right here. And they almost look like fingerprints to me. They look like swirls going around in a circle. So this is one laminated corpuscle, and here's another, kind of this swirl structure. And so remember that laminated means it's in a bunch of layers of connective tissue surrounding a nerve ending. And it's in the dermis, and this is going to be responding to the sense of vibration. And then for the taste buds, strange place to look for it here, but here's the tongue. And so this is the outer surface of the tongue. This is what your food sits on. And so each of these little peg-like structures here is a papilla. So we have several papillae, if we're talking about plural. And then this groove in between each papilla, the food that you eat has to be dissolved with the saliva, and it has to go down into these grooves so that they have access to the taste buds, which are kind of these little almost like grape-like structures here on the sides of each papilla. So you see there are several taste buds here, several taste buds on this portion, and those are on larger structures called the papilla. So on the tongue we have structures called papillae, and the papillae are the structures that have taste buds, and those are these little grape-like structures. Alright, I hope that helps you understand the histology of some of the nervous system structures.